Hi, so today my presentation is going to be about photosynthesis. So there are two stages of photosynthesis. There are light reactions that occur within the thylakoid membrane and the Calvin cycle, which occurs within the stroma. Light reactions occur to convert solar energy into chemical energy of ATP and NADPH. The Calvin cycle occurs to reduce carbon dioxide into sugar with the help of ATP and NADPH from the light reactions. Light reactions are considered light-dependent reactions because they depend on the sun for it to process. However, the Calvin cycle is considered light-independent because it doesn't depend on the sun for it to occur. So the roots of a plant absorb water, and that water is brought into the leaves. And CO2, carbon dioxide, enters the leaf through the stomata, and it's where oxygen exits as well. There are little pores within the leaf. And the sun contributes to this process because it gives off photons that's absorbed into the plant by chlorophyll, which is pigment within the plant, and that is found within the thylakoid membrane. So now the linear electron flow starts, which is what I have right here. A photon energizes the first chlorophyll, and the energy transfers to other chlorophylls until P680 is activated. P680 is known as photosystem 2, and photosystem 2 does function before photosystem 1. And an excited electron from P680 is then taken in by the primary electron acceptor, also known as P680+. In order for P680 to settle back down and gain back that electron that it lost, water is split into three parts: hydrogen, oxygen, and electrons. Now P680 plus is reduced to P680, the oxygen leaves the stomata, and hydrogen ions are then released into the thylakoid membrane right there. Electrons that go through the electron transport chain falls from photosystem 1 to photosystem 2 with the help of the cytochrome. The cytochrome helps transfer those electrons to and from. And from this, energy is being released by pumping protons into the thylakoid membrane, and those protons are the hydrogen ions. This is also known as a concentration gradient. Within this gradient, potential energy is being stored. This energy creates ATP. In photosystem 1, also known as P700, photons excited as well, and one excited electron is lost to the primary electron acceptor, just like within photosystem 2. More electrons go through the electron transport chain, falling from PS1, photosystem 1, to protein ferredoxin, which is FD right here. NADP plus catalyzes with those electrons, reducing NADP plus to NADPH. So now that you have ATP and NADPH, the Calvin cycle can now begin. The Calvin cycle has 3 phases, and the first one is carbon fixation. So it starts off with RUBP, a 5-carbon molecule, also known as a CO2 acceptor. It takes in a carbon, and it turns into a 6-carbon molecule. And the 6-carbon molecule breaks into 2 molecules right away, and these are called 3PG, which is 3-phosphoglycerate, and ATP is being used here. Now, the second phase is reduction, and that ATP being used in the first phase causes a phosphate to be added to 3PG. And this molecule is now called 1,3-biophosphoglycerate because one phosphate is being added to the first carbon of the molecule, and another phosphate is being added to the third carbon of the molecule. Electrons and hydrogen are then added to 1,3-biophosphoglycerate and becomes G3P, which is also called glyceraldehyde 3-phosphate, and this is only half of the glucose molecule. The numbering system throughout the cycle is 3, but just for the sake of the presentation, I had it simplified. So normally you would start off with 3 RUBPs instead of 1. And since that's the case, 9 molecules of ATP and 6 molecules of NADPH are needed to create 6 G3Ps. However, only 1 gets to leave the cycle, and that's because 5 G3Ps are needed to regenerate 3 of the RUBPs, which is what you started with. And if you were to use all of the G3Ps, it'll shut down the cycle, which means no sugar will be created. So therefore, you need 5 to regenerate, and that only leaves 1 to actually leave the cycle, and that 1 is the sugar being created. So that is my presentation on photosynthesis. Thank you.